Okay. So the cell membrane protects what? The ICF, right? The intracellular space from what? Extracellular, right? Space. It's like a boundary, okay? So, and if it's made up of lipid bilayer, which means not everything, right, can go through. Does that make sense? Not everything can go through the cell membrane. Only molecules that are what? That are lipid soluble, right, can freely go in and out. Molecules that are water soluble cannot just enter the intracellular space or cannot leave, right, the ICF, right, into the ECF. There's a boundary. So take for instance electrolytes, ions. They can't leave, just leave in and out through the membrane, right? Freely. They can't. We're looking at lipid soluble water soluble molecules, either hormones, proteins, anything. They cannot freely go in and out. Yeah. So why is that important, right? The first thing, right? The first reason that is important, if you look at it physiologically, is that we can use that to create what? The gradient, concentration gradient, where we can have certain molecules higher, right, on one side of the membrane than the other. For instance, potassium is higher where? In the cell, right, than the outside of the cell. Sodium is higher on the outside than inside the cell. Or else you were frozen for a while. Did you get everything I said? Okay. So the membrane creates a gradient, which means molecules are going to be higher on one side than the other side. Physiologically speaking, right, is that gradient good or not? Is it good thing to have sodium higher on one side than the other, or to have potassium higher in the side than the outside? What do you think? Physiologically speaking, is that a good thing? Yes, it is a good thing. It is. Because we use the gradient to do what? To drive physiologic processes, right? We use the gradient to drive physiologic processes. Take an example. How do cells generate resting membrane potential? Is the movement of potassium, right, out of the cell passively, passive diffusion of potassium out of the cell. Will that be possible without having that gradient where potassium is higher in the cell than the outside? No, it won't. All right. So it's a beautiful thing to have that. Take another example, transport cells that play a role in transportation, the transportation specialists in the body, enterocytes, and the renal cells. How do they do that? How do they absorb dietary nutrients, electrolytes? How does the kidneys reabsorb filtered water, sodium, potassium? They use the gradient generated, right, by just having a cell membrane, either passive transport, active transportation, right? Neurons. How do neurons and muscles become excitable, depolarize? Sodium can move in to increase the membrane potential, right, and depolarize them. Direct action potential, still due to the what? That gradient that we have. So, structure, function. Why is the body organized the way it is? Why do we have cell membranes? Why do we have compartments? One of the reasons is to generate gradients. Why are gradients important? To drive physiologic processes, basically. Now, imagine, right? Next question is to consider, if I have a gradient and I activate a particular physiologic process, what happens to the gradient? So imagine a cell is generating resting membrane potential, right? And potassium keep leaving the cell. What happens to that gradient eventually if that process continues in perpetuity? That gradient will do what? Dissipate. Make sense? Yeah. Oh, I have sodium gradient and the neurons are depolarizing in perpetuity. They keep getting activated and activated and activated. Sodium keeps entering over time. What happens to the gradients eventually? It's going to dissipate, disappear. So it's going to get to a point, if those things continue in perpetuity, it's going to get to a point where they're going to stop. All the physiological processes in the body will stop. And death ensues in that case. So the question is, how do you maintain that gradient? That's the question. We need a way, right, to make sure we maintain the gradient so that we can continue those physiological processes, right, forever, as long as we're alive. Do you know how we do that? Maintain the gradient? Sodium gradient, potassium gradients. How do we maintain it? We use we use transporters. That's why we have what? That's why we have what? Sodium potassium ATPase pump, which makes that pump what? One of the most important pump in your body. That is physiology. If you understand that concept alone, you can understand a lot of processes in the body. Why they are the way they are. Why we need ATP in the first place. That pump maintains the gradient, without which there will be no physiologic processes. So, almost any pathology affecting the cell in the body, almost any, if not all of them, they are going to mess up that pump somehow. And that's why our physiological processes become what? Deranged. Suddenly, I can't think fast enough because my neurons can't fire, depolarize. Suddenly, I have muscle weakness because my muscles can depolarize. Yeah, my cells start misbehaving because almost every pathologic process, whether infectious, non-infectious, ischemic, epoxy, they're going to mess up that pump directly, indirectly, and the body cells will be deranged. Yeah, start misbehaving. People fall ill. Yeah. All right. So sodium potassium ATPase is one active, right? Active, primary active transporter. So basically, it's pumping sodium and potassium against their concentration gradient. It's going to be bringing more potassium into the cell where potassium is already high, and it's going to be taking sodium out of the cell where sodium is already high. So it requires a massive amount of ATP. It will not, at this point now, it won't shock you, right, to realize that sodium potassium ATPase can use up to 1/3, sometimes up to 2/3 of the total ATP made in the cell. It uses up to 1/3 in some cells, and even in some other cells up to 2/3. Imagine how high that ATP is of the total ATP made in a cell. That's a lot of ATP. Okay, so still on this subject, right? Another way I can look at cells, just to understand, sodium potassium is more. Another way you can classify cells in the body, right, is you can classify cells into specialized and non-specialized cells. Because this also allow you to understand why some cells are more susceptible to disease than others. Specialized cells are cells that have extra function. They are above average cells. You can see them as maybe A students. They are above average because they have extra function. Non-specialized cells are every average cells in the body. Yeah, every other average cell in the body will be non-specialized. They don't have any special function. They are just, cells. That's it. So, example of specialized would be what? The muscles, neurons, the renal cells, enterocytes. Those are specialized cells because they have, in addition to being just cells, they have extra function they do in the body. Specialized function that is limited to just those cells alone. No other cell does it. Hepatocytes would be an example, also under the GIT. They are specialized cells, special function. Neurons, muscles, cardiac cells, etc., they depolarize. They are excitable. That's an extra function. Renal cells, they transport stuff. That's an extra function. Enterocyte, transportation, extra function. Hepatocytes, metabolism, transportation, right? extra function. Yeah. So they are specialized cells. Non-specialized cells are going to be other cells in the body. Now, specialized cells, because they have extra function beyond generating just resting membrane potential, they have more, their sodium potassium ATPase pump has higher activity than non-specialized cells. Because now, the gradients I'm generating, they use it for an extra function, which means they need to generate more gradient, which means their sodium potassium ATPase works more, which means it also requires more energy. And that's why those cells use 2/3 of their total ATP production to power sodium potassium ATPase pump, including some other active transporters like calcium ATPase, also to maintain or generate calcium gradients. That'll be another example of a pump that will use massive amount of ATP. Yeah. While non-specialized cells, they only need the gradients for maybe resting membrane potential and some other little things, but no extra function. They will use maybe 1/3 of the ATP, right, to power sodium potassium ATPase just to stay alive. That's it. But they have no extra function. So diseases, pathologies that will decrease ATP in the body, which almost every disease does somehow, whether directly or indirectly. But specifically, we're dealing with hypoxia. Patient doesn't have oxygen. There's no ATP. Patient is hypoglycemic. No ATP. Oh, patient has mitochondrial disorders. Yeah, patient has some sort of toxins that's damaging mitochondria. Patient has carbon monoxide poisoning. Anything you can think of that will limit ATP production will affect what specialized muscles first and specialized cells first before non-specialized cells. So it's not a mistake, right? It's not a coincidence that what mitochondrial disorders are what they present with neurologic and muscular symptoms. Because those cells are special, they need a lot of ATP. So if there's no ATP, they fail first before any other cell in the body. It's not a mistake that conditions that also mitochondrial disorders and other conditions that affect muscles, they die of heart failure, respiratory failure, because all those things require massive amount of ATP, more than specialized cells. So they fail first. Make sense? Okay. Still on the cell membrane. Imagine for some reason no ATP. Sodium potassium stops, ATP stops working. Calcium ATP stops working. What will happen in the cell in that case? What happens? What will be the change in electrolyte concentration in the ICF and ECF you're going to see in that case?
You said that it's not working.
Yeah, the pump stopped working.
So there would be no outward movement of the ion.
So of which ion, if sodium potassium stops working, which ion will accumulate in the cell?
Sodium is in.
Sodium, right. Sodium will accumulate in the cell, correct? Potassium will accumulate outside because normally potassium is brought in, right? So sodium accumulates in the cell. Calcium ATPase to normally pumps calcium out. So if it stops working too, calcium accumulates, right, in the cell. Okay. And what will happen to the cell? So sodium, calcium are accumulating in the cell. What would that lead to?
Swelling.
Swelling. What will follow? Good. And eventually the cell swells up, and that's what we call what? Hydropic change, right? In pathology. A cell is ischemic. Early on, you're looking at histology, the cell swells up, got an hydropic change. That is the mechanism of hydropic change or cell swelling when there's cell injury. Now, if this goes on, there's no correction of the disease. There's no restoration of ATP production. The pump is still down. This goes on for hours. What happens eventually to the cell? Keeps swelling up until what? It ruptures, right? Cell rupture. Now, when the cell ruptures, cell membrane ruptures, disintegrates. The contents of the cell that are normally right protected, right, from the ECF will do what? They're gonna leak out of the cell, right, into the ECF. The clinical significance of that is we can use those contents, those intracellular contents that are now in the serum, in the blood. We can use them as a marker for what? Cell damage, for cell injury. Because we know they should not be in the blood. As long as the cell membrane is intact, those things should not be in the blood. They are in the cell. So I shouldn't see them in the blood. But when I see them in the blood, I can conclude, I can go back and ask myself, what is happening at the cellular level? Oh, the membrane is disintegrated. That's the only reason why those things were appearing in blood. And not only can we use them as markers of cell injury, we can also use them, also some of them, to specifically localize where the injury is. Right? That's why you're using like troponin, right? And CKMB, right? To tell me if there's muscle damage, cardiac muscle injury, MI. Because if there's damage to the muscles, the cell membrane disintegrates, those proteins that are intracellular normally, right, and enzymes will leak out into the blood, and I'm going to see them there. We use creatine kinase to tell us if skeletal muscles undergo rhabdomyolysis, rupture their membrane. We can use AST, ALT, right, to tell me if the liver is damaged, because those are intracellular enzymes. If they're high in the blood, I know, wow, the liver. Oh, ALP to tell me if the biliary tree, the cells of the biliary tree are damaged. Yeah. So that is the concept. So they're going to give you that big picture. Patient has MI, we see those enzymes in the blood. Oh, patient has rhabdomyolysis, we see creatine kinase. Patient has liver damage, you see ALT. But they're going to bring it back to the cell. So you now ask yourself, what is going on in the cell? Why are these guys in the blood? You have to remember, oh yeah, the cell membrane. It's gone. That's why they're in the blood. Just the cell membrane. Just one tiny pump, right? Tiny cell membrane. Look at how many clinically relevant, right? Concept linked to that. And that's how you master your exam. You have to be able to go macroscopically as well as come back down microscopically, right, to the cell and figure out what is going on at that level in this disease or in this pathology or in this state. Still on cell membrane. So we know. Let me link something here. Since we're on general parts now, because of the necrosis and cell death. Apoptosis, right, is different, right, from necrosis. One of the major differences is that the cell membrane is intact throughout apoptosis. In apoptosis, like auto-digestion of the cell is being digested inside, but we won't know on the outside because the cell membrane remains intact throughout that process. But in necrosis, we're going to know on the outside because the cell membrane will disintegrate and the content that should not be in the ECF, right, will be in the ECF, and that's how the body would know that a cell is undergoing pathologic death or necrosis. But in apoptosis, the body won't know. We won't know if our cell is undergoing a programmed death because the cell membrane is intact throughout. Yeah, the process. And that is why apoptosis will not trigger acute inflammation, but necrosis does. Yeah, apoptosis will never trigger acute inflammation, but necrosis does. Why? Because acute inflammation is what? The cell's response to injury. That's what inflammation is. Acute inflammation is the body's response to cell injury. So the body can't respond to injury if it doesn't know, right, that there's death in the first place. So in apoptosis, there's no way to know. Every content is in the cell membrane. There's no spillage, nothing, no inflammation. But in necrosis, the body will know because of spillage of intracellular content. And that's why once there's necrosis, what will follow necrosis is inflammation, acute inflammatory response, damage control, immune cells respond, going there, trying to contain the damage, kill whatever is causing the damage. Use an infectious agent, yeah. Destroy cells that are dead already, get them out. Clean up the mess so that we can heal. Yeah. So cell membrane. Okay. We mentioned, right, that since cell membrane is a lipid bilayer, only certain molecules can cross, right, the cell membrane freely without any help. Yeah. And why some molecules cannot, so they require, like the ions will require transporters, either passive or active transporters. Yeah. Carrier proteins, etc., for those that are not lipid soluble. But for those that are lipid soluble, they can cross freely. And this applies on so many levels. So if you go to endocrine system, where we have hormones, this concept will help you understand why certain hormones, neurotransmitters, signaling molecules, they behave differently. Why some have receptors where on the membrane and some have receptors in the cell. It has to do with their property, the property of the hormone itself, or the property of the signaling molecule. Because you can't change this property of the cell membrane in a normal physiologic state. The property of the cell membrane is the same no matter what. But the proteins that will interact, or molecules that will interact with the cell membranes, they have different properties. So those that are lipid soluble, hormones that are lipid soluble, molecules that are lipid soluble, which includes hormones that are made in what? Adrenal cortex, all of them, made from cholesterol, they're all lipid soluble. Thyroid hormone, vitamin A and D, they behave like steroid. Yeah, lipid soluble vitamins. Those guys, they can cross the cell membrane and enter, right, the cell. So they have intracellular receptors as well as intranuclear receptors. While those hormones or neurotransmitters or molecules that are water soluble, that are polar molecules, they cannot cross the cell membrane, so they require what? A membrane receptor that would transduce, right, the signal into the cell. Right. So any hormone that is not on the list of the lipid soluble ones, you consider them, right, as being water soluble, and they must have what? Membrane receptor. Insulin, glucagon, epinephrine, norepinephrine, etc. Yeah. So which ones of those two categories of receptors or hormones with their receptors, membrane receptor or the ones with the intracellular receptor, which ones we use the GQ system, the G system of proteins, or GQ, GI, GS, receptor tyrosine kinase, Janus kinase? It must be what? The ones that have membrane receptors. The ones that use those G receptors. The mechanism of the intracellular, right, receptor is what? Since the receptors are going to end up in the nucleus for the intracellular receptors, so they're going to end up in the nucleus, the only thing they can regulate in the nucleus is DNA. That's the major content of the nucleus. So the only thing that can regulate in the nucleus is DNA. So the mechanism of action of intranuclear receptors is gene regulation or regulation of gene expression. They regulate transcription. That's what they do. So they are basically what? Transcription factors. That's what they are. So they can bind to DNA, which means they have a DNA binding motif like zinc finger, leucine zipper. Yeah, to bind to DNA. Most of the steroid hormones and stuff, they use zinc finger, except cyclic KMB, which is leucine. And you're gonna be asking me, oh, Emmanuel, you just said intranuclear receptors, isn't cyclic KMB part of the GS pathway, right? Which is the membrane receptor, right? Yeah. So the second messenger for GS, which is cyclic KMB, it can work in the nucleus and in the cytoplasm. So this is an exception you have to know because they can test that. The second messenger for the GS receptor. GS receptor itself is on the membrane. But once you activate the receptor on the membrane, we activate adenine cyclase, which will make cyclic AMP. Cyclic AMP can activate protein kinase A in the cytoplasm. It can also enter, cyclic AMP can also enter the nucleus and bind to its own transcription factor in the nucleus and regulate gene expression in the nucleus using leucine zipper to bind to DNA. Yeah. Other.
So basically, CIP is a steroid hormone, not the GS receptor.
So okay, yeah. So cyclic AMP itself, right, is a steroid, right? It has steroid properties. Cyclic AMP, it can behave like steroid hormones. Although it's just a second messenger, but it can behave like steroid. Good. But the receptor itself, GS receptor, is on the membrane. But once I activate the receptor and I make cyclic AMP can enter the nucleus. Not the GS receptor, it's cyclic AMP itself only that can enter, right? The nucleus, yes. And that's important because if I ask you, a patient has, you ask a question that they're asking you, how will glucagon turn on gluconeogenesis? Right. So patient has been fasting for 2 days and the serum glucose is still normal. And the question will be asked, right? So we know at that point, two pathways that will be responsible for maintaining serum glucose at that point are gluconeogenesis and glycogenolysis, right? I break down glycogen to glucose. I synthesize new glucose, gluconeogenesis. How will glucagon activate those pathways? Especially gluconeogenesis. Two things. It's going to do two. It's going to be two ways. Although glucagon itself is what is it uses a GS receptor, which is on the membrane. Is a lipid, is a water soluble, right? Peptide. So it's going to have a membrane receptor, which is a GS receptor. Once glucagon binds to that membrane receptor and turn on adenylyl cyclase and cyclic AMP goes up, so cyclic AMP will activate for protein kinase A. And protein kinase A can phosphorylate, right? The already made enzymes in the cytoplasm and turn on, right, gluconeogenesis and glycogenolysis. The second thing cyclic AMP will do is what? Enter the nucleus, bind to its transcription factor receptor, and turn on the genes to synthesize what? More of those enzymes, right? So it can activate already made enzymes in cytoplasm through phosphorylation. It can also turn on the gene, right, to make more of the enzymes in the nucleus. Make sense? Okay. So that. And if you're asking you any of those thyroid hormones, how do they work? Yeah. They enter the nucleus, turn on the gene, right, for whatever they want to do. How will cortisol increase glucose? enters the nucleus, turns the gene for gluconeogenesis enzymes, makes more liver makes more glucose. How will vitamin D increase calcium absorption? Goes to the enterocyte, cross their membrane, enters the nucleus, turn on the gene for cal binding, calcium binding protein. Calcium will be absorbed more. Yeah. How does thyroid hormone increase metabolism and stuff? Yeah, same thing. Enters the nucleus, turn on the genes for the metabolic enzymes, turn on the genes for sodium potassium ATPase pump. Yeah, all those things will increase oxygen demand, ATP use, ATP production, metabolic rate goes up. Yeah, so they work right that way. So that one, right, for cell membrane also, for sure. Excellent.
Yes.
Next organelle. So that's cell membrane, basically. Just some basic principles that we're going to be seeing through the system, right? That make, oh, even before we leave the cell membrane, also the same principle of being lipid soluble and co also applies to drugs, by the way. Don't forget that. Applies to medications. Yeah. So medications are, and you get the property of a drug determines the volume of distribution of that drug. Basically, how much volume in the body, right, would the drug occupy? How much of the compartment we have would the drug occupy? So will the drug be stuck only in the plasma, in the blood, or can it occupy both plasma and the interstitium, ECF, or can it actually cross the cell membrane, right, and end up in the ICF and occupy the whole total body water? So it depends on the property of the drug. Drugs that are lipid soluble, they can cross everything. They can leave the blood vessels, cross the cell membrane, and occupy all the compartments. So they have very, very high volume of distribution. Drugs that are water soluble, that are protein bound, they're gonna occupy just either only the blood vessels, right? Plasma, they can't leave. Or at most they occupy the plasma and the what interstitium, right? Only the ECF, they can't cross the cell membrane, right? So they would not enter the ICF. So they have smaller, right? Volume of distribution. So your question can give you the VD of two drugs and ask you how are they different. I'll give you the VD of a drug and ask you what is the property of the drug. So the VD is equals to plasma volume, approximately 5 liters or so. And you're thinking, okay, this is a likely water soluble, it's dissolved in only plasma, it's protein bound, it's stuck in that compartment. If the VD is like up to 40 liters, which is like the whole total body water, then you know it's what